First of all, I welcome you all for Emerged classes. Here you can emerge through education. In this segment, let us talk about the structure of embryo sac. Here, this embryo sac represents the female gametophyte. It represents female gametophyte. This female gametophyte is developed from one functional megaspore. It developed from single functional megaspore, and it is haploid in nature. In an ovule, embryo sac is present below the nucleus. It is situated below the nucleus. At maturity, an embryo sac contains 7 cells and 8 nuclei. A matured embryo sac is 7-celled and 8 nucleated. In an embryo sac, antipodal cells, egg operators and polar nuclei is present. Embryo sac contains egg operators, antipodal cells and polar nuclei. In this first one, egg operators. In an embryo sac, the 3 cells which are present towards the micropylar end, they are the egg operators. The cells which are present towards micropyle, they are the egg operators. Out of these cells, central one is called egg and other 2 cells are called synergids. Here, the central cell is the egg and remaining cells are synergids. In the synergid, special cellular thickenings or finger-like projections are present known as filiform operators. In synergid, special cellular thickenings are present called filiform operators. ಈ ಫಿಲಿಫಾರ್ಮ್ ಆಪರೇಟರ್ಸ್ ಕೆಲಸ ಏನು ಅಂತಂದರೆ ಇಟ್ ಪ್ಲೇ ಆನ್ ಇಂಪಾರ್ಟೆಂಟ್ ರೋಲ್ ಇನ್ ಗೈಡಿಂಗ್ ದ ಪೋಲನ್ ಟ್ಯೂಬ್ ಟು ಎಂಟರ್ ಇನ್ ಟು ದಿ ಓವ್ಯೂಲ್. Filiform operators guides pollen tube to enter into ovule. Next, antipodal cells. These are the cells which are present towards the chalazal end. 3 cells present towards the chalazal end, they are the antipodal cells. Next, polar nuclei. This central large portion of the embryo sac contains polar nuclei. These two polar nuclei later fuse to form a central cell, and it has a nucleus called diploid secondary nucleus. Later it fused to form central cell, and it has secondary nucleus, and its ploidy is diploid. Why it is diploid? Polar nuclei is haploid, so n plus n is equal to 2n. ಸೊ ಯಾವಾಗ ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ ಫ್ಯೂಸ್ ಆಗುತ್ತೋ ಆವಾಗ ಒಂದು ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಆ ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಪ್ಲಾಯ್ಡ್ ಏನಾಗಿರುತ್ತೆ ಅಂದರೆ ಇಟ್ ಈಸ್ ಆಲ್ವೇಸ್ ಡಿಪ್ಲಾಯ್ಡ್. ದಿಸ್ ಸೆಕೆಂಡರಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಈಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಡೆಫಿನೆಟ್ nucleus. So this is all about the structure of embryo sac or female gametophyte. Now let us see some of the differences between microsporogenesis and megasporogenesis. What is microsporogenesis? It is the process of formation of microspores. Megasporogenesis means formation of megaspores. ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ನಲ್ಲಿ ಮೈಕ್ರೋಸ್ಪೋರ್ ಫಾರ್ಮೇಷನ್ ಆದರೆ ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್ ನಲ್ಲಿ ಮೆಗಾಸ್ಪೋರ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಎರಡನೇ ಡಿಫ್ರೆನ್ಸ್ ಏನು ಅಂದರೆ the site of microsporogenesis is anther. Microsporogenesis process ಎಲ್ಲಿ ಕಂಡು ಬರುತ್ತೆ ಅಂತಂದರೆ it takes place in anther, whereas megasporogenesis it occurs in ovule. Next difference ಏನು ಅಂದರೆ we know that during microsporogenesis 4 microspores are formed. In a similar way ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಪ್ರೊಸೆಸ್ಸಲ್ಲೂ ಕೂಡ 4 ಮೆಗಾಸ್ಪೋರ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಬಟ್ ಒಂದು ಮೇಜರ್ ಡಿಫ್ರೆನ್ಸ್ ಏನು ಅಂತಂದರೆ ಇನ್ ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಆಲ್ ದಿ 4 ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಆರ್ ಫಂಕ್ಷನಲ್. 4 ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಆರ್ ಫಂಕ್ಷನಲ್. ಬಟ್ ಇನ್ ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಔಟ್ ಆಫ್ ದಿ 4 megaspores 3 megaspores degenerates and only 1 megaspore remains functional. Here 3 megaspores degenerates and only 1 remains functional. Next, ಇನ್ನೊಂದು ಡಿಫ್ರೆನ್ಸ್ ಏನು ಅಂತಂದರೆ ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ನಲ್ಲಿ ಮೈಕ್ರೋಸ್ಪೋರ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ ಅಂದ್ವಿ, ಹೌದಲ್ವಾ? ದಿಸ್ ಮೈಕ್ರೋಸ್ಪೋರ್ ಹೆಲ್ಪ್ಸ್ ಇನ್ ದಿ ಡೆವಲಪ್ಮೆಂಟ್ ಆಫ್ ಮೇಲ್ ಗ್ಯಾಮೆಟೋಫೈಟ್. ಮೇಲ್ ಗ್ಯಾಮೆಟೋಫೈಟ್ ಆ್ಯಂಡ್ ಈ ಮೇಲ್ ಗ್ಯಾಮೆಟೋಫೈಟ್ನಲ್ಲಿ ಏನು ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ? ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ವೆರಾಸ್ ಇನ್ ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್ megaspores helps in the formation of, it helps in the development of female gametophyte, and in female gametophyte egg or female gamete is formed. So this is the major difference between microsporogenesis and megasporogenesis.